Embryologists play a crucial role in understanding the intricate processes involved in the development of various organ systems. As an embryology professor, I have the privilege of teaching medical students about the fascinating world of embryogenesis. Today, we will delve into the detailed mechanisms and pathways that govern the development of the hindgut and its associated structures, with a primary focus on anorectal development and the formation of congenital anorectal malformations. Hindgut development is a complex process involving morphological changes and differentiation. The sonic hedgehog signaling pathway, expressed in the endodermal epithelium, regulates cell fate, proliferation, and differentiation through homeobox genes and transcription factors. The role of sonic hedgehog signaling in anorectal development is crucial. Expression in the hindgut endodermal epithelium stimulates mesenchyme development and induces the urorectal septum, a key step in cloaca division. PBX1, a transcription factor regulated by contributes to anorectal specification by interacting with mesenchyme and endodermal epithelium. Overall, the development of the rectum relies on intricate interactions between mesenchyme and epithelium. Signaling pathways, such as the FGF pathway, and cellular communication through integrins and cell adhesion molecules are essential for proper morphogenesis and tissue remodeling. In summary, the formation of the urorectal septum is influenced by a combination of genetic and environmental factors. Mutations in certain genes and exposure to teratogenic substances can disrupt the development of this critical structure. The formation of the cloacal membrane is critical for proper anorectal development. Defects in cloacal membrane development can lead to various congenital anomalies, known as cloacal malformations. The etiology of cloacal malformations can be multifactorial. Genetic factors, including mutations in HOX and BMP signaling pathway genes, have been implicated. Disruptions in retinoic acid signaling can also contribute to these anomalies. Clinical implications of cloacal malformations vary depending on the severity and associated structural abnormalities. Early diagnosis and appropriate surgical management are essential for optimizing outcomes in affected individuals. Anorectal malformations are often influenced by genetic factors. A better understanding of the contributing genes and their mechanisms of action is crucial for unraveling the underlying etiology. Numerous genes have been implicated in anorectal malformations, including Krebs binding protein, CBP, FREM1, and CHD7. These genes play significant roles in embryonic development, particularly in the formation of the hindgut and anorectal structures. The mechanisms of action of these genes involve the regulation of cell proliferation, migration, and differentiation. Their dysregulation can disrupt the delicate balance of hindgut morphogenesis, leading to anorectal malformations. In summary, homeobox genes are crucial for normal anorectal development. They exhibit specific expression patterns and have important functional roles in specifying cell types and maintaining anorectal structure. Dysregulation of these genes can lead to anorectal malformations. Rectal rotation and fixation are critical processes in the establishment of proper anorectal orientation. Molecular regulation, including signaling pathways and biomechanical forces, are integral to these processes. One key signaling pathway involved in rectal rotation is the transforming growth factor beta, TGF-beta, signaling pathway. TGF-beta ligands secreted by the epithelium and mesenchyme regulate the migration and colonization of the rectum during rotation. Biomechanical forces, such as gut peristalsis and differential growth rates, also contribute to rectal rotation and fixation. The coordinated action of these forces ensures proper alignment and anchoring of the rectum. The WNT signaling pathway is crucial for the development of the anal canal. Components such as beta-catenin, GSK3-beta, and axin regulate cell fate determination. Dysregulation of this pathway can lead to anorectal malformations. Sacral development abnormalities are frequently associated with anorectal malformations. Understanding the embryological basis and clinical correlations provides valuable insights into the diagnostics and management of these conditions. The development of the sacrum involves the complex interplay between mesodermal and ectodermal tissues. Anomalies in mesodermal segmentation, neural tube closure, or neural crest migration can negatively impact sacral development. Clinical correlations between sacral anomalies and anorectal malformations suggest shared molecular and genetic predispositions. The identification of specific genetic markers and diagnostic tools can aid in early detection and intervention. The development of the rectum and anal canal exhibits interesting evolutionary aspects across different vertebrate species. Comparative anatomical studies and phylogenetic analysis shed light on the diversity and conservation of these developmental processes. Comparative anatomical studies reveal variations in the morphology and development of the rectum and anal canal. Phylogenetic analysis helps trace the evolutionary changes and molecular adaptations associated with these structures. Understanding the evolutionary aspects of rectal and anal development provides a broader perspective on the variability and plasticity of these developmental processes and their clinical implications in human medicine. Congenital anorectal malformations require surgery to improve long-term outcomes. Techniques like SARP and LARP are commonly used. Advancements in minimally invasive approaches and tissue engineering can enhance surgery success and precision. The study of embryology provides fundamental insights into the development of the rectum and anus. Understanding the complex processes involved can help us diagnose, treat, and even prevent congenital anorectal malformations. This knowledge is crucial for aspiring medical professionals.